Step 1 of phosphorylation of glucose. In the first step of glycolysis, a phosphate group is transferred from ATP to glucose, making glucose 6-phosphate. The glucose 6-phosphate is more reactive than glucose, and the addition of phosphate also traps glucose into the cell since glucose with phosphate cannot readily cross the membrane. This reaction is catalyzed by the enzyme hexokinase. Step 2 of isomerization of glucose 6-phosphate. Glucose 6-phosphate is converted into its isomer, fructose 6-phosphate. This reaction is catalyzed by the enzyme phosphoglucose isomerase.
Phosphofructokinase. In the third step of glycolysis, phosphofructokinase, the addition of phosphate from ATP to fructose 6-phosphate forming fructose 1,6-biphosphate. This is irreversible, highly spontaneous reactions is also the rate-limiting step of glycolysis, meaning it is lowest and most regulated step. It often involves magnesium to weaken bonds for phosphate release. Aldolase catalyzed. In step 4 glycolysis, aldolase splits fructose 1,6-biphosphates into dihydroxyacetone phosphates and glyceraldehyde 3-phosphates. Only glyceraldehyde 3-phosphates can proceed, so the cell uses thiosyphosphates isomers to convert dihydroxyacetone phosphates into glyceraldehyde 3-phosphates. The dihydroacetone phosphate is called DHAP and the glyceraldehyde phosphate is called G3P.
The power of triose phosphate isomers. Inside every cell, countless chemical reactions drive life forward. One such reaction is catalyzed by a key enzyme, triose phosphate isomers, or TIM. It plays a crucial role in glycolysis, helping convert sugars into energy. TIM uses its active site to rearrange DHAP into G3P, ensuring this essential reaction occurs rapidly and centrally. By catalyzing this reaction, TIM helps regulate the flow of metabolites through glycolysis. Both DHAP and G3P are essential intermediates leading to energy production in various biosynthetic pathways. Glyceraldehyde 3-phosphate dehydrogenase is a crucial enzyme in the glycolytic pathway, responsible for the conversion of glyceraldehyde 3-phosphate into watery-based phosphoglycerate. This reaction is vital for energy production and is coupled to the reduction of NAD+ to NADH. In this process, G3P reacts with NAD+ and inorganic phosphate. The enzyme facilitates the oxidation of G3P, resulting in the formation of 1,3-biphosphoglycerate, a high-energy compound, while NAD+ is reduced to NADH. The enzyme's active site facilitates the transfer of electrons from G3P to NAD+, producing NADH. The addition of Pi creates 1,3-bisphosphoglycerate, an intermediate with high energy, essential for subsequent AT generation and glycolysis.
Hello everyone, my name is Kenneth Dial and I will be discussing the 7 and 8 steps of glycolysis. In step number 7 and 8 of glycolysis, we see crucial transformations that help convert glucose's energy into a usable form. So, we'll start in step number 7, which is the phosphoglycerate kinase. Here, it involves the enzyme phosphoglycerate kinase, which plays a key role in ATP production. Here, 1,3-biphosphoglycerate or 1,3-BPG, it is generated in the previous step and it donates a high-energy phosphate group to ADP, resulting in ATP, the energy currency of the cell, along with 3-phosphoglycerate, a lower-energy molecule. This process, known as a substrate-level phosphorylation, allows the cell to recover some energy previously invested, yielding 2 ATP molecules for each glucose molecule processed. Moving forward to step number 8, phosphoglycerate mutase. In here, the enzyme phosphoglycerate mutase facilitates a molecular rearrangement by relocating the phosphate group from the third carbon of 3-phosphoglycerate to the second carbon, forming 2-phosphoglycerate. This is a next preparatory step for the next energy-generating reaction in glycolysis, positioning the substrate for optimum energy release. Together, these steps illustrate how glycolysis effectively converts the energy stored in glucose into ATP, supporting various cellular processes.
Enolase is a reversible reaction. So, first, the substrate of 2-phosphoglycerate will bind to the active site of enolase enzyme. 2 of the magnesium ion are present in the active site. So, this ion will bind to and interact with oxygen atom of the carboxyl group of the structure 2-phosphoglycerate. This interaction ensures proper alignment of the substrate and prepares it for the catalytic reaction. a lysine is present in the enzyme active site. It will remove hydrogen from carbon number 2 from this structure. So by removing hydrogen from this structure, 2-phosphoglycerate, it will form a new structure known as enolate and ion intermediate. The oxygen from this structure is seen to have a change of positive ion to negatively ion charge. Thus, this reaction has high energy and it is in a negatively charged state. This is the time where magnesium ion will interact with the negatively charged oxygen atom to make the reaction stabilize. So in organic chemistry, we combine positive and negatively charged ion. It will result in an equal reaction. The last reaction involves glutamine to donate hydrogen to the hydroxyl group. Together, they will form what we call H2O or water. And it can be released from this structure. So the dehydration process results in the formation of double bond between carbon number 2 and carbon number 3 to produce what we call phosphoenolpyruvate. This is pyruvate kinase. So phosphoenolpyruvate, or PEP in short, and ADP bind to the active site of pyruvate kinase. Inorganic cations like potassium and magnesium are essentials for this process. So they bind to negatively charged residues in the active site, helping to stabilize the reactants and position them for the reaction. So pyruvate kinase transfer the phosphate group from PEP to ADP. So this forms ADP, or the energy currency of the cell, and leaves behind pyruvate. So the phosphate transfer is spontaneous because phosphoenolpyruvate has a very high energy level compared to the adenosine triphosphate. So this large energy releases and it drives the reaction to go forward. So after the phosphate group is removed from the phosphoenolpyruvate, it will become an unstable enol form of pyruvate. This enol form spontaneously converts to the more stable keto form of pyruvate, so without requiring additional energy.
So in this lesson, we are going to discuss about the pyruvate pathways. So as you can see in the illustration, let's just have a quick refresh about this one. So when you start with glucose and ultimately form pyruvate downstream, that is glycolysis, shown in red. If instead you move in the reverse direction, starting from pyruvate and going up to glucose, that is gluconeogenesis, shown in the blue. So after the quick refresher, we can actually do the pyruvate. So this is the most important molecules in any of these biochemical pathways because pyruvate can at any point in time change itself or transform itself through a chemical reaction into one of 4 different products. So in all of these next examples where we are going to go through 4 different biochemical pathways, so we're gonna start with the reactant of pyruvate and ultimately be able to form 4 different end products. The first pathway that pyruvate can take is to turn itself into lactic acid, or also known as the lactate. And about the pathway referred to as anaerobic glycolysis. Now, what does anaerobic means? So, it means in the absence of oxygen, and aerobic, there is no oxygen. This is glycolysis occurring without the presence of oxygen. Now, normally oxygen is usually important in the ability to conduct glycolysis. So recall that glycolysis and all metabolic pathways have one goal in mind, and that is to work together to form cofactors and reactant that can be used with one another to ultimately send everything to the electron transport chain to generate a massive amount of ATP. So that is the job of biochemical reactions occurring in the body. So we have to remember that oxygen is the ultimate or the final electron acceptor in the electron transport chain. it should come as no surprise to you that in the absence of oxygen, which is to say that the electron transport chain will simply not work. So there is no point in going through regular glycolysis in this case. So it should make a little bit of sense why instead of going from glucose to pyruvate and then sending pyruvate to the TCA cycle, we're just going to convert pyruvate to lactate acid, and that's done in the anaerobic glycolysis pathway, which is going to be the first pyruvate pathway that we will be talking about. So these are the actual purpose of these reactions in the body. So the conversion of pyruvate to lactate acid is what's termed anaerobic glycolysis, and the enzyme that does this reaction is called the lactate dehydrogenase, or LDH for short. This enzyme is active in certain conditions. Again, as I've mentioned, this is anaerobic glycolysis, so it should come as no surprise that the main condition in which LDH is active is in low oxygen state. Now, clinically, low oxygen state occur in things like infection, hypoxia, ischemia, and heart failure. So we obtain lactate levels when we test people's blood, because an elevated lactate is telling us that the patient does not have adequate oxygenation, and there might be some underlying infection or some underlying hypoxic or ischemic event going on. Now, in certain tissues, this is the pathway that pyruvate will primarily go through, and those tissues are shown. Here, the testis, the lens, white blood cell, the cornea, the medulla of the kidney, and the red blood cells. And for this one, the cofactor in the reaction catalyzed by lactate dehydrogenase is vitamin B3. So maybe you will ask why we want to turn pyruvate into alanine, and the reason is something called the Cahill cycle. So pyruvate is not able to jump between muscle and liver, so it's simply unable to do it. So in order to recycle carbons between the muscle and the liver, we actually have to disguise pyruvate as alanine or glucose to be able to make this a possibility. So I want to show you some biochemical pathways. So let's start in the muscle. So starting with pyruvate, you can turn pyruvate into alanine using the ALT enzyme that I've shown you on the previous slide. This conversion will also use up glutamate and spit off alpha ketoglutarate. These are cofactors, so it's pyruvate plus glutamate goes to alanine plus alpha ketoglutarate. Since pyruvate cannot jump between muscle and liver, alanine can. That is why we convert the pyruvate into alanine. Once you have the alanine can go, once it is from the muscle into the liver, can be converted back into pyruvate using alpha this well in the Cahill cycle. We actually have and the splitting off and glutamate. So it's the same reaction in the muscle, you already learned. Backwards.